Welcome to our video lecture on membrane structure, IB Biology topic 1.3. Our essential understanding for today's lecture: the structure of biological membranes makes them fluid and dynamic. Fluid, they're not solid, they're not stuck in place. Dynamic, they change a lot. They are constantly changing. Our objectives for today's lecture: we are going to observe the fluidity of the membranes. We're actually going to watch a couple of videos that show us just how much membranes can move around. We're going to outline the development of membrane models. We haven't always known as much as we know today. Technology hasn't always been as good as it is today. So we definitely have changed our minds about what membranes look like over time. We're going to talk about the functions of membrane proteins, and then we're going to talk about the function of cholesterol, and that'll be our day. These are some of the vocabulary words that you probably already know. If you want to pause the video and look up some of these, you certainly can. This is your video lecture. If you want to keep going, I'm going to talk about a lot of these, all of these actually, in the lecture, so you can review that way, if not right here. But definitely activate the knowledge that you already have. We're going to build on that. All right, so here is a lovely diagram of a plasma membrane. There's a lot of stuff. It looks super messy and chaotic. There are so many pieces and parts. And we'll talk about the pieces and parts in detail more, but let's look at some of those chunks already now. So we have peripheral proteins. These are proteins that are stuck just on one side or the other side of the membrane. That's different from integral proteins, which are sometimes also called transmembrane proteins. And these guys go all the way through from one side of the membrane to the other. We definitely have our phospholipid bilayer. So we've got our phosphate heads, lipid tails on one side, and then we have phosphate heads and lipid tails on the other side. So that's why we call it our phospholipid bilayer. There's one layer, two layers that orient toward each other. We also have some cholesterol. There's some cholesterol molecules in here. Cholesterol, usually we think about it as being bad for our health, but we have to have it in our cell membranes in order for those membranes to function. We also have some proteins that will have some chunks of carbohydrates stuck to them. So the part that I've colored red here is the carbohydrate part. The blue is the protein part, and we put them together, we have a glyco, which is the carbohydrate. Protein, that's the protein. Lots of super cool stuff in our membranes. All right, so we're going to watch this video on fluidity of the lipid bilayer. The scientists are literally going to take tiny tweezers and pull the cell membrane out, like pinch it out.
To demonstrate the remarkable fluidity of the lipid bilayer, a piece of the plasma membrane of this neuronal cell is picked out with laser tweezers. The membrane is pulled out into a tube, and the tube is moved around.
Cool beans, right? So that neuronal cell, that's the same thing as a nerve cell. So that was a neuron. And that membrane clearly is kind of stretchy and fluid. We're going to watch another video here. So in this video, they're going to take some membrane and they are going to label it with some fluorescent additions. So they're going to make the membrane glow in the dark. The proteins in the membrane are going to glow in the dark. But then they're going to bleach out the glow-in-the-dark part of the proteins. They're going to watch to see how the bleached part, does it stay in the same place or does it move around? So this is called FRAP, fluorescent recovery after photo bleaching. I know my little thing is in the way. Fluorescence recovery after photo bleaching. This is so cool.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
In the video, they mentioned GFP. GFP, that stands for green fluorescent protein. And we're basically taking proteins and adding a little piece that is green fluorescence. So it glows in the dark. We steal that green fluorescent gene from jellyfish because that's what we do in science. We borrow from jellyfish. And so you could see in the first piece of the membrane that the glow-in-the-dark piece is definitely filled in the gaps, so those proteins were moving around a lot. In the nuclear membrane, the second part of that investigation, those guys, there was really little to, if any, movement. So membranes are fluid, but not equally fluid. Some are super, super fluid. All right. So we're going to talk about some of these little details, lots of pieces and parts, some good vocabulary in here. So our membranes are composed of phospholipid bilayers. These are some big, some big lovely pieces of those phospholipids. So we have the hydrophilic head. So this is that phosphate group. So that's why it's phospho. And then this is the hydrophobic tail. This is the lipid part. The phosphate head is hydrophilic. That means it does like to hang out near water. It will dissolve in water. The tails are hydrophobic. They will not interact with water. So they'll not dissolve or hang out. So they definitely are repelled by water as opposed to the phosphate head, which is attracted, which is super cool. So this one molecule is both hydrophilic and hydrophobic at the same time. And so we say that it is amphipathic. Amphi is a prefix that means both, like amphibians can live in both land and on water. And then path is feel. So these molecules feel both hydrophilic and hydrophobic. Definitely make sure that you know phosphate heads, hydrophilic, fatty acid tails. And there are two fatty acid tails, 2 fatty acid tails. These guys are hydrophobic. So what's kind of cool about our amphipathic phospholipid bilayers is that they can build these beautiful little bubbles. Sometimes we call these bubbles micelles. And then these micelles, we think, got a little bit bigger and started holding in some cool stuff. And those micelles eventually evolved into plasma membranes. Soap bubbles will also form these micelles because a soap molecule has usually some kind of sodium or potassium ion in one end, which is hydrophilic, And then those fatty acid chains after that, lots of carbons and hydrogens, which is hydrophobic on the other side. So just be aware when you are purchasing face soap and you're like, ooh, micellar face wash, that sounds fancy. Yeah, it's just soap. Don't pay too much money for your soap. Good, good. All right. So how do we know? How do we get to our ideas of the phospholipid bilayer, our fluid mosaic model? Well, a long time ago, 100 years ago, these scientists decided that there had to be a bilayer just because of the interactions that the bilayers could do with water inside and outside. So there had to be a bilayer. We had to have some way of interacting with water inside the cell and outside the cell. This is how we came up with our bilayer idea. In the 1930s, two scientists, Davison and Danieli, they came up with this idea that we had our bilayer was here. So our phospholipid bilayer was here. And then that bilayer was sandwiched between two layers of protein. We knew that there had to be proteins in the cell membrane. We could actually see them with our very first electron microscopes. And so Davison and Danieli were like, all right, we've got two layers of protein, kind of like bread, surrounding our phospholipid bilayer on the middle, and they called it their sandwich model. And it was a really good model. It was a good idea considering the data and the technology that we had at that time. But then these guys, Singer and Nicholson, did something super cool. So here is a cross section through a plasma membrane with an electron microscope. So it kind of makes sense. We can see protein, phospholipids in between. So Davison and Danieli's model made sense. What Singer Nicholson decided to do was they took the cell membrane and they froze it. And then with a super, super skinny, tiny knife, they actually cracked the membrane in half. So they kind of, if we're using Davison Danieli's model, they opened up the sandwich. They peeled back the two phospholipid bilayers. And what was expected if I had lipids, protein, lipids, well, lipids are like butter. And so when we opened it up, it should have been smooth like butter. But instead, there were all these crazy divots. So many little divots. And so Singer Nicholson said, well, sandwich doesn't make sense. Look at all those divots. So many chunks. Should have been like, the whole thing should have been like this, smooth like butter. But instead we've got all these holes. And this is why they decided that we have, instead of sandwich protein, lipids in the middle, we have a fluid mosaic model instead, where the proteins are actually embedded within and on the edges of our phospholipid bilayer. Then we got some more details later. We started talking about like where things are hydrophilic and hydrophobic in the proteins, and are they barrels or are they alpha helices, and lots of more detail came about. But this fluid mosaic model is still the accepted model, our best explanation for the structure of a membrane. And this is just part of the nature of science. Science is always improving, and that doesn't make science bad or wrong that we changed our minds. But that we are always looking for more information. We're always trying to improve our technology, make better observations, come up with better theories. And so the nature of science, it's pretty common that we falsify theories by getting better information and better theories. So we were able to falsify that Davison Danieli model because of that freezing and cracking open of the cell membrane. We were able to see that those proteins were more globular, not fibrous. We'll talk about these kinds of proteins more in our next unit. And then very recently, we could use our green fluorescent protein tagging, adding those glow-in-the-dark pieces to the proteins in the membrane, and we can literally watch them move around in the membrane. Good. All right, so membrane proteins. We've talked a lot about there are proteins in the membrane. There are 6 different functions, and you do need to memorize these functions. So one of the things that membrane proteins that are stuck in membranes, one of their jobs is intercellular joinings. And so they can hold together two different cells. In the skin, in other parts of connective tissue, we want stuff to be stuck together. And so these proteins can help to make that happen. Some of our proteins in membranes act as enzymes. They help to catalyze chemical reactions. They help to break things down or build things up. And when we get, again, to our next unit, when we're talking about photosynthesis and respiration, we're going to talk a lot about proteins that are enzymes in our cell membranes and in other membranes within the cell. Our membranes can be transport, active transport, passive transport. Remember that active transport requires energy in the form of ATP. Passive transport, no ATP required. It just happens, which is pretty cool. Cell-to-cell recognition. Recognition is another one of our membrane protein functions. This is super common in the immune system. So our bodies need to know what cells belong and what cells do not. And so lots of communication can happen because cells can recognize each other as belonging or not belonging. Our immune system can do its job. Another job of membrane proteins is to anchor or attach. There are lots of things that are stuck to cells. Here I've got some more proteins. I have actin filaments that make our cytoskeleton. Actin is another kind of protein. So here we've got a protein attached to more protein. So pretty cool stuff going on there. And then this last thing, signal transduction. This is super cool. So we've got here some kind of hormone or neurotransmitter, some other kind of chemical messenger. And it's going to bind to a receptor, which is a protein in the cell membrane. And it's going to cause some kind of shape change in this protein. And then the shape change is going to cause something else to happen on the inside. The hormone itself never actually gets into the cell. It just does something to the protein, and the protein sends the signal to the inside. That's signal transduction. It's a lot of things to remember. If only we had a mnemonic device that could help us. Oh, but we do. So if you haven't already visited BioNinja.com. BioNinja is out of Australia. It is amazing, an amazing resource. Definitely hang out on that website lots of times. They have a brilliant, clearly brilliant, look at this guy, mnemonic device to help us remember membrane protein functions. And it is JET-RAT. Junctions, enzymes, transport, recognition, attachment or anchorage, and transduction. JET-RAT. All right, so we're going to talk a little bit, and I mentioned this at the beginning of the slideshow, positions of membrane proteins. Where even in or through or on the membranes do we find our proteins? So two kind, we have integral, which we sometimes call transmembrane, and then we have peripheral. Integral proteins, transmembrane proteins, they're going to go from outside the cell all the way through to inside the cell. So they're going to go all the way through the membrane from one side to the other. Our peripheral proteins are going to stay on the outside, like your peripheral vision is your vision along the outside of your field of vision. Peripheral proteins do not go all the way through the membrane. They just sit on one side. They might go into the middle, but they do not go all the way through from one side to the other of the phospholipid bilayer. One of the cool things about our integral proteins especially, so some of them act as channel proteins, and this is for passive transport. So we know that our fatty acid tails here in the middle, they are hydrophobic, which means that things that are polar or have a charge like ions cannot get through here. They will be repelled by these hydrophobic interactions amongst the fatty acid tails. But sometimes we need to get sodium into and out of cells, especially if you're a nerve cell. Nerve cells depend on lots of sodium and potassium and chloride ions. And we need to get these guys in and out of cells. And so what we do is we send them through these channel proteins. What's kind of cool is how the proteins, the amino acids in the protein sort themselves as they're getting built by the ribosomes. All of the polar amino acids are going to be toward the center of this channel because sodium is very polar and polar things and other polar things like each other. And so sodium can easily pass through this polar channel. I'm going to erase some of my scribbles because that's a lot of scribbles. So our sodium can pretty easily go through the inside of this polar channel. But remember that our phospholipid bilayers, they are hydrophobic. They are very non-polar. And so we can't have polar amino acids for this protein hanging out next to our non-polar hydrophobic fatty acid tails in the middle of our phospholipid bilayer. And so what happens is this amazing thing. So I said that our polar amino acids sort themselves on the inside of the channel. Non-polar amino acids are going to sort themselves on the outside of the channel. So we have non-polar out here, and then we have polar on the inside here. So our sodium can get through, but the protein can still pretty happily sit next to our hydrophobic fatty acid tails, and that is amazing. All right, talking a little bit more about the positions of our membrane proteins and how they can act as receptors, recognition. So it's been a COVID life, my friends, and so I thought I would bring in some COVID proteins. So this is a simplified version picture of a COVID virus. It's got its RNA, single-stranded RNA here. That's what the SS is, single-stranded RNA. It does have a little bit of a lipid membrane, and then it has some membrane proteins, and then it has these guys, spike proteins. And the spike proteins are what give COVID its name. We call them coronaviruses because these guys in our electron microscope pictures look a little bit like a crown around the virus particle. So these spike proteins, these glycoproteins, and here's a more detailed picture showing like amino acid bubbles poking around. So this guy enlarged is this guy. And what it can do is actually bind to this other protein right here called ACE2. And this stands for angiotensin. Oh, I don't remember. Angiotensin something enzyme. And this is on a lot of our lung cell membranes, and it helps us manage our blood pressure. So this smelly virus has built these proteins, and these proteins will bind to these other proteins that are in our bodies, and that's how the RNA of the virus can sneak through this ACE2 receptor and into our lung cells, and that's why people often have respiratory issues if they have COVID. What our messenger RNA vaccines, what these guys are doing is we're actually getting in the vaccine just a little piece of messenger RNA that tells our bodies how to make just the spike proteins. So our bodies, our ribosomes will make spike protein. And the spike protein will sometimes bind to the ACE2 receptor, but there's no viral RNA. We only have the RNA for the proteins. We don't have the rest of all of this stuff. And so our bodies don't make the virus. We only make the spike proteins. And our immune system will then see these spike proteins that our cells are making, and our immune system will have a reaction, and we will bind to and remember how to kill off this virus. So that's how the messenger RNA vaccines work. It's all about these proteins, which is super cool, these membrane proteins. All right, enough of a tangent there about COVID. Let's practice a little bit. Which structure is an intrinsic or integral protein? Remember, this is the same thing as transmembrane. Transmembrane means it's going to go all the way across or through the membrane. Hopefully you're thinking the answer is 3, 2, 1, B. Because B goes from one side all the way through. That's going to be our transmembrane protein. Since we're here, though, let's go ahead and label all the other guys. What is A pointing to? That's going to be the phosphate head of a phospholipid. And remember that phosphate heads are hydrophilic. The inside is hydrophobic fatty acid tails. What about C? What do we think C is pointing to? This little guy right here. That is cholesterol. We'll talk about that a little bit more in a couple slides. And up here we have all these little hexagons stuck to this guy. This is a protein. This is a carbohydrate. And when we put the two together, we get a glycoprotein. Glycoproteins are especially important for recognition. So when we talk about like our blood types, we're talking about glycoproteins. All right. So cholesterol. What even does cholesterol do? Why is it a good thing to have in our membranes? It helps us regulate fluidity of the membrane. It's pretty cool. It also is hydrophilic, but just a little bit hydrophilic. Most of it is hydrophobic. All this green part is what's stuck in between the fatty acid tails, the hydrophobic fatty acid tails of the phospholipid bilayer. And that little piece sticks out between the phosphate heads. Cholesterol helps us regulate membrane fluidity. So if our membranes get too hot, they start to melt like butter. That's bad. We don't want our cell membranes melting. And so when we get a little bit too hot, cholesterol is going to help to hold things together so that we don't melt. When we get too cold, the membranes could freeze. We don't want that to happen either, because then they get kind of cracky and they crystallize, and that's bad. Cholesterol will help to keep things from freezing. So cholesterol is like the mama bear. Everything was, no, baby bear. Baby bear was, everything was just right. Not too cold, not too hot, not too fluid, not fluid enough. Cholesterol helps to keep everything right in the middle. All right. It reduces fluidity, but can also increase fluidity. It's going to reduce the permeability to small polar molecules. So it's going to keep sodium and potassium from trying to sneak through. It is going to separate tails to prevent crystallization when it gets too cold. And sometimes it'll also help to hold on to those peripheral proteins, the proteins that only are on one side or the other side of the membrane. Cholesterol is good, but not too much cholesterol, just enough. And that, my friends, is the end of our lecture on membrane structure. So hopefully you were amazed and astounded by FRAP and the GFP proteins in the membrane floating around. Thinking about development of membrane models, we need to know Davison-Danieli was the sandwich model, and then Singer-Nicholson is better. Singer-Nicholson replaced Davison-Danieli. Singer-Nicholson is our fluid mosaic model. Mosaic, good. We can describe the functions of membrane proteins. Remember, jet, rat. junctions, enzymes, transport, recognition, attachment or anchorage, and then signal transduction. And then the function of cholesterol is just going to regulate the fluidity of our membranes. Good work today, my friends. See you in class.